Hi friends, welcome to my channel. Today we are looking into the topic cell division mitosis. Let's look into some important definitions. Cell. It is the structural and functional unit of all living organisms, and all cells originate through division of pre-existing cells. Cell division. The process by which a parent cell divides into two or more daughter cells is known as cell division. Mitosis. The process in which one cell gives rise to two identical daughter cells is known as mitosis. Meiosis, also called reduction division. The process in which one parent cell gives rise to four daughter cells, each with half the number of chromosomes of the parent cell, is known as meiosis. Mitosis. Mitotic cell division was discovered by Fleming in 1879. Term first used by also Fleming in 1882. Here we see mitotic cell cycle. It contain four main stages. Those are G1 phase, S phase, G2 phase, M phase. In G1 phase, protein and RNA synthesis occurs. In S phase, DNA synthesis occurs. In G2 phase, protein and RNA synthesis occurs. Now look into different stages of cell cycle. In mitotic cell cycle there are mainly two stages. First one is interphase, and the second one is M phase. In interphase, there are three substages. Those are G1 phase, S phase, G2 phase, and in M phase there are two stages. First stage is mitosis. In mitosis, prophase, metaphase, anaphase, telophase, and second stage is cytokinesis. Detail explanation of different stages of mitotic cell cycle. Interphase. Interphase of the cell cycle is a contain of three distinct stages. G1 phase. First intermediate gap stage in which the cell grows and prepares for DNA replication. S phase. Synthesis stage in which DNA is replicated. G2 phase. Second intermediate gap stage in which the cell finishes growing and prepares for cell division. In interphase, DNA is present as uncondensed chromatin. Here DNA not visible under microscope. DNA is contained within a clearly defined nucleus. Centrosomes and other organelles have been duplicated. Cell is enlarged in preparation for division. M phase. The period of the cell cycle in which the cell and contents divide to create two genetically identical daughter cells. This phase is comprised of two distinct stages. Mitosis. Nuclear division, whereby DNA is separated into two identical nuclei. Cytokinesis. Cytoplasmic division, whereby cellular contents are segregated and the cell splits into two daughter cells. Adhere while explaining. Look into diagram shown below simultaneously. Prophase. Chromosomes become increasingly shorter and thicker due to increase in condensation. The spindle apparatus is not yet organized. Chromosomes are comprised of genetically identical sister chromatids. The nuclear membrane breaks down and the nucleus dissolves. Metaphase. Microtubule spindle fibers from both centrosomes connect to the centromere of each chromosome. Microtubule depolymerization causes spindle fibers to shorten in length and contract. This causes chromosomes to align along the center of the cell, equatorial plane or metaphase plate. Anaphase. Continued contraction of the spindle fibers causes genetically identical sister chromatids to separate. Once the chromatids separate, they are each considered an individual chromosome in their own right. The genetically identical chromosomes move to the opposite poles of the cell. Telophase. Once the two chromosome sets arrive at the poles, spindle fibers dissolve. Chromosomes decondense. Nuclear membranes reform around each chromosome set. In cytokinesis. After anaphase, carbohydrate-rich vesicles form in a row at the center of the cell, equatorial plane. The vesicles fuse together and an early cell plate begins to form within the middle of the cell. The cell plate extends outwards and fuses with the cell wall, dividing the cell into two distinct daughter cells. Because this separation originates in the center and moves laterally, it is described as centrifugal. In mitotic cell cycle there are 3 checkpoints. These checkpoints check the cell capable of divide or not in different stages cell cycle. Those are G1 checkpoint. Determine appropriate growth conditions, sufficient nutrients, cell size, presence of growth factors, etc. Assess level of DNA damage, from ionizing radiation or UV. G2 checkpoint. Determine state of pre-mitotic cell, suitable cell size required for successful division. Identify and replication faults, changes to DNA sequence will distort genetic fidelity in daughter cells. Metaphase checkpoint. Ensure proper spindle assembly and correct attachment to centromeres, prevents non-disjunction events. See here in diagram G1 checkpoint present at here. In this checkpoint if the cell grow enough the cell enters into S phase otherwise goes to resting stage. Okay guys, hope you get everything. See you all in next video to get further notifications. Please like, share and subscribe.